Proteins, the unsung heroes of cellular dynamics, shape our biological world with their intricate structures and complex functions. These molecular marvels serve as architectural alchemists, weaving the fabric of life with an exquisite blend of science and art. Their magic lies in their ability to fold into unique tertiary structures through non-covalent interactions, including hydrogen bonds, van der Waals forces, and electrostatic interactions. This folding process, akin to an origami masterpiece, forms the foundation of the diverse and dynamic architecture of cellular components. Each protein, with its unique shape and structure, holds a specific role in the grand scheme of cellular processes. Some act as gatekeepers, others as messengers, while some take on the role of builders and fixers. Their function is dictated by their structure, and any change in this structure can drastically alter their role within the cell. Without these architectural alchemists, the intricate architecture of our cells would be a mere fantasy. Proteins also conduct the symphony of life as catalytic maestros, orchestrating biochemical reactions with remarkable specificity. Imagine proteins as conductors, leading an orchestra of enzymes, each playing its part to create a harmonious melody of life-sustaining reactions. Enzymes, a special class of proteins, are the workhorses of the cell, catalyzing biochemical reactions with finesse and precision. They accelerate these reactions, making them happen at a pace that sustains life. But how do they do this? At the heart of each enzyme is the active site, a specialized region defined by its precise three-dimensional structure. This active site is where the magic happens. It orchestrates the binding of substrates, the molecules that enzymes act upon, and facilitates their transformation into new products. Now let's delve a bit deeper into the mechanisms of catalysis. One of the ways enzymes facilitate chemical transformations is through acid-base catalysis. Here, the enzyme donates or accepts protons, driving the reaction forward. The second mechanism is covalent catalysis. In this, the enzyme forms a temporary covalent bond with the substrate, lowering the energy barrier of the reaction. This bond is then broken, releasing the product and leaving the enzyme ready to catalyze another reaction. Finally, there's metal ion catalysis. Some enzymes employ metal ions to stabilize negative charges on a substrate, or to generate a more reactive form of the substrate, thereby accelerating the reaction. Each of these mechanisms is like a different instrument in our enzymatic symphony. Some enzymes may use just one mechanism, while others might use a combination, depending on the nature of the reaction they are catalyzing. The result is a symphony of reactions, each enzyme playing its part, each reaction occurring at just the right time and in just the right way. It's a display of biochemical harmony led by proteins, the catalytic maestros. Thus, proteins conduct the enzymatic symphony, playing the role of catalytic maestros. Imagine a world where proteins communicate, transmit signals, and respond to changes in cellular environments. Welcome to the world of dynamic allostery. In this realm, proteins are not static entities, but dynamic performers that flexibly adapt to the ever-changing cellular stage. Allostery represents a fascinating aspect of this dynamism. It's the process by which proteins transmit effects from one site to another, often situated far away within their structure. Picture it this way: allosteric proteins are like intricate musical instruments. When a musician, let's call them a ligand, plays a note or binds to a specific site, the instrument doesn't just produce a singular sound, it resonates, creating vibrations that alter the entire instrument's structure. This is similar to how ligand binding induces conformational changes in proteins, influencing their activity at distant sites. But what's the significance of this musical metaphor? Well, it's in these subtle shifts and changes that proteins regulate key cellular processes. They control signal transduction, where cells receive and process information from their surroundings. They regulate metabolic pathways, ensuring cells efficiently convert nutrients into energy. And they mediate protein-protein interactions, forming complex networks that underpin myriad cellular functions. This dynamic allostery is akin to a symphony, where each protein performs its part, responding to cues and contributing to the overall harmony of cellular functions. Through dynamic allostery, proteins contribute to the harmony of cellular functions. In the intricate dance of cellular life, proteins choreograph the formation of supramolecular complexes. Just as dancers form intricate patterns on the stage, proteins engage in a dance of their own, forming complex structures through protein-protein interactions. These interactions are mediated by specialized domains within the proteins, such as SH3, PDZ, and leucine zippers. Like choreographers guiding their performers, these domains direct the proteins to engage with one another, forming intricate supramolecular complexes. Now, these complexes are not just for show. They play pivotal roles in orchestrating various cellular processes. For instance, they are instrumental in the conduction of signal transduction cascades, a series of events that allow cells to respond to their environment. They also choreograph the movement of vesicles within the cell, a process known as vesicle trafficking. Even the replication of DNA, the very blueprint of life, is overseen by these supramolecular complexes. The precision in these interactions is astonishing. Proteins must come together at just the right time, in just the right place, and in just the right way. Any misstep in this intricate dance can disrupt cellular function, leading to disease. So the next time you think of a dance, remember the proteins, their complex choreography, their precise movements, and their integral role in the dance of life. The choreography of these protein complexes is vital for the smooth functioning of cells. In the cellular symphony, proteins also play the role of conductors, orchestrating post-translational modifications. Now, what are these modifications? They are essentially molecular tailoring that happens after the protein has been synthesized. They involve the addition of chemical groups like phosphate, glycan, or ubiquitin to specific amino acid residues within the protein. Imagine a musician tuning their instrument before a performance. The instrument is already built, but it needs fine-tuning to perform at its best. Similarly, these post-translational modifications fine-tune proteins, adjusting their activity, stability, and localization within the cell. But why is this important? Well, this molecular tailoring helps proteins to adapt to their roles, allowing them to perform their functions with precision. They influence a variety of cellular events, from cell signaling, which is like the cell's communication system, to gene expression, the process by which information from a gene is used to create a functional product like a protein. Moreover, post-translational modifications also help cells respond to environmental cues. It's like the conductor adjusting the orchestra's tempo in response to the audience's reaction. These adjustments help the cell to adapt and respond to changes in its environment, ensuring the smooth performance of the cellular symphony. Through these modifications, proteins conduct the symphony of cellular dynamics. They ensure that each protein is perfectly tuned and ready to play its part in the grand performance of life. Proteins exist not as rigid structures, but as dynamic ensembles, adopting multiple conformations. This concept, known as the conformational ensemble, provides a fascinating dimension to our understanding of proteins. Rather than being static, proteins are in a constant state of flux, transitioning between different states. So, why is this important? Well, this dynamism allows proteins to interact with various binding partners, each interaction guided by a different conformation. Let's imagine a protein as a talented dancer. This dancer doesn't stick to just one routine, but can switch between different dance styles, each style corresponding to a specific partner. In the same way, proteins can alter their structure to match the requirements of different binding partners. This adaptability is a major asset in the cellular world. It allows proteins to respond to changes in their environment and to fulfill multiple roles. For instance, a protein might bind to one molecule to carry out a specific function, then change its conformation to interact with a different molecule for another function. This continuous switching between different conformations creates a dynamic flow, a choreography if you will, within the cell. Each protein, with its ensemble of conformations, performs its part in this intricate dance, ensuring the smooth running of cellular processes. Hence, the conformational ensemble is a crucial player in the choreography of cellular dynamics. In the cellular cosmos, proteins form intricate networks, establishing the basis for cellular communication. These networks, known as protein-protein interaction networks, are a marvel of biological engineering. Each protein, like a node in this network, collaborates with others to facilitate a myriad of cellular processes. From signaling cascades that transmit information like relay runners in a race to metabolic pathways that convert food into energy, these networks are the lifeblood of cellular dynamics. They also play a crucial role in regulatory processes, ensuring that each cellular function is carried out at the right time and place. It's like an intricate dance where each protein knows its partner, its steps, and its rhythm. However, these networks are not static. They adapt and evolve, responding to the ever-changing cellular environment. Understanding these interaction networks is key to unraveling the orchestration of cellular events by proteins.